undergraduate and graduate teaching. He has been a fellow of the Institute of Physics, London, England since 2004. So please, let's give him a warm welcome and take it away. Hello.
Thank you. I hope everyone can see my my screen. Yes? Can you?
No longer.
No.
Let's see. Okay, so let me make sure that I'm sharing my screen properly. Give me a moment to do that. Okay, here we go. And okay, now you can see it?
Yes.
Okay, great. All right. Okay, so the title of my talk is From Living Organisms to Living Machines. So let me give you an outline. I want to start by talking about what I think about as the borders between the living and non-living, which is relevant to our meeting. And I also want to talk about the great shapers of biological cells and organisms, evolution, development, plasticity. And to try to make this as concrete as possible, I'm going to do a thought experiment with you, and we will together think about something I'm going to call a cell machine. It will have, perhaps that's too grandiose, we'll see that it has certain limitations, but it may also get us thinking about how one would take these ideas and implement them for artificial devices. And in order to understand more about control for such a device, I will also talk a little bit about creating biologically inspired neural networks. And then I will briefly talk about some of the new directions. Where most of the people here are more focused on going from the artificial to the biological or being inspired by biology, but biologists are actually going in the sort of the opposite direction, trying to create artificial organisms. And so I'll briefly talk about synthetic biology and artificial life. And as we start to blur these borders and are successful in doing so, I think we're going to have to also deal with some of the social and ethical considerations that this raises. And then I will summarize and hopefully reach some interesting conclusions. If someone has a question as I talk, you're welcome to ask, but I will also hope to leave some time for questions afterwards. And my hope is to really, this is sort of a, as opposed to being a talk where I focus a lot on my own previous work, this is a talk where I'm hoping that I can get the whole group thinking about some of these fundamental questions with me. So our conference is called Living Machines, and it raises two very fundamental questions. The first question is, are living cells and organisms machines? Is that a fair way of describing them? And then, are there any human-created machines that are alive? Now, I recognize that these questions might seem almost too philosophical, but I hope to convince you during the course of the rest of this talk that these are actually useful questions to answer, and they both help us understand more about living things at the same time as they perhaps inspire us to create novel artificial devices. There's a very interesting and thoughtful essay that was recently published by Cornish, Bowden, and Cardenas, and they compare and contrast several possible theories. I'll just mention them, I won't go into them in detail. There's the work of Maturana and Varela in the 70s on autopoiesis, self-creation. There's the work of Eigen and Schuster on hypercycles. There's the work of Rosen on what he calls MR systems. There's the work on autocatalytic systems, and there's work of Ganti on what he called the chematon. And so they go through these, and I will say more about what they derive from all of them. But at the outset of their essay, they make some very important points, which I think I'll start to sort of set the framework. The first point they make is that no matter how one defines the machine, whether a very simple tool, such as an axe, a more complicated machine, such as an airplane, or even an entire factory, it remains true that the machine does not make itself or maintain itself. It needs the input of an external agency in order to do this. I think someone may need to mute themselves because I'm starting to get echoes. Roger, that may be you. Okay. And the second point they make is that living organisms are quite different from machines. They depend on their environments for material, food, and they release material, such as CO2, water, and other excreta into their environments. So they are thermodynamically open systems. That is, they take sources of energy, They maintain internally low entropy, high structure, but they release material that has much higher entropy, so thermodynamics is not violated. But they are organizationally closed systems. We'll say more about that in a little while. And all of the catalysts they need, that is all the structures internal to them that allow them to transform, for example, energy sources or materials that they themselves are manufacturing or receive from other cells, are closed systems. That is, they make and they maintain all of those catalysts internally. And all the catalysts they need, apart from some metal ions, they make themselves. Okay, so this in a certain sense throws down the gauntlet because it tells us that if we claim that something is a living machine, we have to argue that somehow it actually has some of the characteristics of living things. So this raises the question, what are the characteristics of living things? And one of the ways that people try to do this, you'll find this in textbooks, is that they list characteristics associated with living cells and with organisms. And it's essentially kind of a checklist. So living organisms and individual cells show internal organization. They have metabolism. That is, they have the network of processes that are capable of taking in certain inputs. I mentioned energy sources. They can transform one set of chemicals into other chemicals, and many of the things they transform the chemicals into are either needed for maintaining themselves or for their function. So for example, if a molecule is involved in generating motion, then there will be the universal energy currency, ATP, will be made available such that those energetic events can occur. So if you have, for example, molecules that can actually walk, the ATP will power that, or you'll have muscle molecules which can seize hold of one another and then move, giving rise to muscle contractions. All of these things are maintained by the internal metabolism. Another critical function is homeostasis. They maintain certain levels of certain chemicals at certain values, and they work either as individual cells or as organisms as whole to stay within certain ranges of acidity, temperature, pressure, and so forth. They're capable of growth, so they can make themselves larger, and they can reproduce. They can make copies of themselves. And finally, they're responsive to the environment. As things change around them, they can change appropriately to what's going on around them. So to try to show both the applications of these ideas, but some of their limitations, let's look at some examples. So if we do so, we'll see that the borders are a little bit fuzzy. So let's start with something that in principle should be very easy. So let's ask the question, is a rock alive? So if you actually look inside the structure of rocks, you see that they have highly organized crystalline regions. Think of a geode, for example. So that's something that actually is highly organized, and crystalline structures in general are highly organized. So that satisfies one aspect of the checklist. And within certain ranges, if you apply force to a rock, you throw it, or you step on it, you throw it into water, it maintains its integrity. In that sense, it has a kind of resistance to change, which you could refer to as a kind of resilience. But in terms of the other parts of the checklist, it certainly doesn't, in general, grow, reproduce, and so forth. So it doesn't satisfy all the aspects of the checklist, but it already shows you that using a checklist is problematic because other natural phenomena have some aspects that are clearly shared with other living things. Let's try a harder case. Is a virus alive? And I just want to say, I'm not going to take a stand on this. I'll point out the reasons why people say yes and people say no. But in the literature, the technical literature, will find that this is actually a debate. Some people say no and some people say yes. And the reason is, and unfortunately, because of our current situation, we are much more familiar with the effects of viruses on human health. So if you've ever seen pictures of viruses in detail, you see that they have highly organized internal structure. The particular one we've been dealing with has these rather beautiful looking spikes on it, which give its name corona. And internally, it also has complex structure, including the messenger RNA that it needs for making copies of itself. However, unlike other living things, you can actually make many numbers of viruses, and you can crystallize them as a powder, and you can put them on the shelf for long periods of time. And the reason you can do that is they can only reproduce by taking over the machinery of a cell. So for example, bacteriophage, they look like a lunar excursion module. They have these little legs. They can land on top of the cell. They can inject their material that hijacks the cell. The cell takes over, making all of these different components, and they self-assemble. The cell dies as a consequence, and it releases very large numbers of the viruses. And a critical aspect, which we are dealing with on a fairly regular basis, is that viruses make poor copies of themselves. So some of the duplicates are much worse than the original virus, and some are actually far better at, for example, evading the immune system or dealing with someone who's vaccinated. So they have this ability to change over time. Are they alive? Well, again, this seems to satisfy more elements of the checklist, but it's still somewhat unsatisfactory. Now, here, we come to something that actually satisfies everything, and it seems to be very helpful to have that. Bacteria have complex internal structures. Bacteria have complex metabolism. They can maintain internal homeostasis. They can grow and reproduce. And some bacteria, for example, have these little flagella, these whip-like structures that allow them to tumble, and they can actually follow gradients. So they can avoid regions that temperature is wrong, or they can follow gradient towards a particular food that they need and ingest it. So they can respond to the environment to survive and find what they need in the environment. And when they reproduce, they do not make perfect copies. And again, that allows them over longer periods of time to change, which is, for example, why we are now dealing with antibiotic-resistant bacteria in many hospitals. Because having found antibiotics that destroy bacteria, some of the bacteria, because they don't make perfect copies, are resistant to those antibiotics, and then those copies are the ones that survive. Okay, so this satisfies the checklist, but let's look at some other interesting cases. If you have a red blood cell after it's born, it loses its nucleus. But for quite a period of time, it's capable of still doing oxygen exchange, of making its way through your blood capillaries and doing its job. It hasn't fallen apart. It isn't, it's still doing its function. So is it alive? It can't reproduce, but it's still doing its functions. Another example that people like to argue with is a mule alive? After all, it's sterile, it can't reproduce. Now, after a certain age, human beings often cannot or do not reproduce. I would be very uncomfortable saying that therefore they're not alive. So if you don't have something, an element on the checklist, does that mean you're not alive? So what this is suggesting is we need a broader way of thinking about living organisms. And so I'm going to return to that review I mentioned. And in reviewing the different theories of life that have been generated, they summarize a series of key ideas that they believe have to be incorporated into any theory of life. And we've mentioned some. I'm going to say a little bit more about them. So the first component is it should be thermodynamically open to allow supply of energy to be harnessed. This is actually fairly critical, as they made the point that living systems are capable of finding energy sources. And those energy sources may not initially be in the form that they immediately need. And so they have some way of assimilating those materials and transforming them into the things that they actually do need. And in part of the way they do this is by having these specific catalysts, these things that can actually transform one molecule into another. In some cases, what they transform are, in fact, themselves can act as catalysts. And it's important that those catalysts only primarily generate the appropriate set of things, because if they start to generate other things, these could be called parasitic reactions, and they could actually suck off some of the material that has to be going through a cycle and render it useless for the cell. we talked about catalytically closed because it has to be the case that all the catalysts that it needs are present and manufactured by it. Otherwise, it's constantly dependent on getting them from the environment. And then we mentioned this idea of being structurally closed. So most cells have a membrane that separate itself from other things that are not itself and from the environment. And this mentions also the importance of concentration gradients associated with these barriers that are used for energy management. A specific example that's very important for the things that I study is the gradient of ions across cell membranes that are used by neurons for signaling. The fact that there's energy used to maintain those gradients is actually used for particular purposes. And of course, things like pumping things into and out of cells require these concentration gradients be properly managed. It's very important that there's a mechanism for storing and reading information. The cells we're familiar with, that's generally DNA, and all of the associated machinery that's involved, molecular machinery that's involved in reading DNA and making it available to generate messenger RNA and then proteins. And then it should be capable of controlled growth. This is one of the points that Cornish, Bowden, and Cardenas emphasize very, very strongly, that if you don't have a way of controlling growth or regulating it, generally at the metabolic level, you have the possibility that many of the things that you generate will start to build up. This is very important. For example, the disease of phenylketonuria occurs because there's a particular enzyme that takes phenylalanine and transforms it into other molecules. And if that's absent, and this is something that a genetic defect that some people have at birth, then the phenylalanine starts building up and none of the subsequent things are made, and that can actually lead to retardation. It can be handled by someone having a very careful diet that is low in phenylalanine. But this gives you an example, a concrete example of how important it is to control the availability of substrate and how fast and how much, not just different components are manufactured, but also the growth of the cells that make up an organism. Okay. Now, what I also want to mention is that there are these major shapers of biological systems of cells and of organisms, and they are evolution, development, and plasticity. I want to do a contrast between them and the way we deal with human-made devices. So in human-made devices, generally, if we're going to make a machine, we work through the design. And if we're going to program something, we often create an outline or a pseudocode version of that program. And that becomes a very important basis for what we do subsequently. One of the important things in human design is that if we make things, we try to make them as uniformly as possible, because we expect that those who are purchasing the things we manufacture are going to want them to all be the same. To give you a concrete example, if you opened up, you ordered from Amazon online a set of 6 glasses, and they came in and every single one was slightly tilted, had imperfections in the glass, discolorations, but they all held 8 ounces. You would probably send them back because you'd be very unhappy. Certainly wouldn't want to set at your table when you had guests with such glasses. But in contrast, if you went into a classroom, said hello, and 30 identical faces turned to greet you, you would be very, very disturbed. So clearly there's a difference between the way we manufacture things and what happens in biology. Finally, of course, it's not enough to simply make things. We have to be able to control them, whether they're complex machines like construction devices, or they may be various kinds of bots that are running in the background as we do our shopping online. All of these things are the way in which we do human-made devices, and the focus is on uniformity and on getting the same results every time if possible. This is radically different than what happens in cells and organisms. We'll say more about the details of this, but organisms are not designed. They evolve, and I'll say more about that. They develop, and that's how you make complex organisms. And they're subject to plasticity. This is very important for how networks of neurons change, but even individual cells are capable of forms of plasticity. And so now what I want to do for several minutes, and I hope you'll tolerate this, is to do a thought experiment with you. I want us to create a device, which I'm going to call, perhaps with a little bit too much grandiosity, a cell machine. And this will help illustrate the ideas in a concrete way. I don't think that right now this could be built, though I might be mistaken. And part of the reason of talking about this at a conference like this is that some people might be inspired by what I say and decide they want to try making it. That would be very interesting. So let's imagine you had a 3D printer that had some additional interesting properties. First of all, let's assume, and this is something that it is part of its specification, it has wheels and grippers so that it can move itself and it can also move other things that it encounters. It's programmable, and it includes a program that describes its own components and structure. It can wirelessly send and receive signals from other similar 3D printers. And finally, it is capable of making 2 copies of itself. And it's also capable of printing structures that are slightly larger than itself because of its mobility. Okay, so I've just given this artificial cell 4 properties. You can imagine we can make 3D printers quite small. Maybe it's a cubic centimeter on the side. Let's see what we could do with something like this. Okay, let's first point out that the importance of being able to make copies of yourself is really quite interesting. Let's say that in an hour it could make 2 copies, and that's all it does. It just makes those 2 copies and then it stops making copies. Well, in an hour, there'll be 3 devices, the original and 2 copies for a total of, so 1 plus 2 is 3. Now in 2 hours, the most recently made 2 copies will each make 2 new copies, so that'll be 4. So the total number of devices will be 1 plus 2 plus 4, or 7 devices, which I point out is 8 minus 1. And so, and you can show this by induction. If someone asks me, I'm happy to do it. You can show that in n hours, there will be 2 to the n plus 1 minus 1 devices. And that means that this is making an exponentially growing number of devices. So that's really quite interesting. Now, just making that many devices is, and again, if you just only could make, if the same printer only made 2 copies every hour, then it would make 2n copies, which is a far smaller number. So the ability to make copies of itself is already very powerful in terms of the numbers. But the other aspect of it that's very important is the motility and the ability to send messages. Because since they can send messages to one another, they can tell each device where to position itself relative to the other devices. So let's take a concrete example of what you could do. Each of the devices could say to another newly created device, line up to my left or line up to my right, and then you would end up with a set of devices in a line. Now you could go further because each device, because it's a printer, could print a surrounding shell which encased it entirely and was made of very stiff material, and that as that material dried, it would adhere to the adjacent devices. The consequence of this is that you could assemble a stiff structural element in parallel relatively quickly. You could think of it as a kind of a bone. Now, if other devices lined up, say above or below this stiff structural element and printed a soft bellows around themselves, and they had the ability to contract and expand those bellows and they attached to one another, then what you could have is the contractile actuator, which also could be assembled in parallel around the stiff structural elements. Now, the interesting consequence of this is, let's imagine you had 2 such stiff elements and you also had devices that printed a joint, like a ball and socket joint. And now you had actuators that connected at the top and at the bottom. And the resulting device, which had all been assembled in parallel relatively quickly because of the exponentially increasing number of elements that were moving into the right place, could move up or down. And so it could be used as a limb or a leg. Now, how would you use this device? Well, one way is you could try to hand design a controller for it. But another way is you could use something called genetic algorithms. So for those who are not familiar, just briefly review, I assume many of this audience have heard of genetic algorithms, but the basic idea is that you have a structure, let's say you have this limb-like structure, and you have some control signals to tell the actuators when to be active and when to be silent, let us say. And you can describe the parameters for that particular set of activations as a list of numbers. You know, time and amount of activation or whatnot. You could write down a whole string of those numbers. Now, what you do is you can start with random values, and you can create a population of strings that have the parameter values in them. And then you drop them either into the device or into a simulation of the device, and you see what it does. And you evaluate it based on whether it actually, for example, rhythmically moves the limb up or down, or even moves it at all. You could set up a sequence of fitnesses, just moving it at all, then moving it up, then moving it down, then moving it rhythmically. And if you did that, what will happen is some of the strings will have higher fitness than others. So in the next population, you take the best string and you might actually replicate it exactly, but the rest of the strings, what you do is, and including the original one, is you make small variations in their values. You could even take two strings, take the beginning of one and the end of the other and combine them that way. And so you have a new population where you've changed some of the parameters and you've built on the ones that are most effective. And the key thing is that the higher the fitness, the more replicates of the original string are present in the new population. So as a consequence, any variation that is successful will be overrepresented in the next generation. Now, if you do that over time, and this is work I did some years ago with Randy Beer, others have done this. We actually were able to show that if you had a locomotion, simple body for locomotion, you could use genetic algorithms, control the parameters for model small neural networks, and you could get this to walk at high speed. So this is clearly a solution that genetic algorithms can solve. Now, you may not want to start with a very long limb. You could start off with a much simpler device, perhaps just 3 of the printers making the structural, or 6 of the printers making the jointed structural element, and maybe 4 or 5 on top and bottom. But the cool thing about a device like this is that once you have it solved for a particular small number of elements, if you want a larger one, all you have to do is to have one more doubling period. And now you have twice as many elements, but since you already have rules for how they should be organized, it's not that hard to see how you could then use this to create much larger devices. Now, I deliberately just focused on one thing. There are many other things you could build with devices like this. If you think about it, only your imagination would limit what kinds of positioning, what kinds of things the cells, these artificial cells could put around themselves, and what kinds of very complex 3-dimensional structures they might be capable of making. I use this as a concrete example, but there are many other possibilities. Now, where did I get all these ideas from? Well, I stole them from biology, because all the processes I've just described are just a simplified explanation of how organisms are shaped by evolution and development. And I wanted to do it in such a way that you wouldn't be put off by using all sorts of foreign terms or seeing these strange looking fluid-like devices making copies of themselves, and you start thinking in terms of actual machines. But in the developmental process, you have a fertilized egg that divides repeatedly. Each cell has the DNA, which is a complete self-description of the organism, like the program that I described that has the self-description of the 3D printer. Cells have receptors for chemicals released by other cells and can release chemicals themselves and can follow chemical gradients. So that's where I got the idea of these wireless signals that the little devices transmit to one another. That's what effectively the cells are doing as a developing organism is being put together. And then I didn't go through this, but you can actually have some cells that die as part of the developmental process. So you would have a case where a printer would essentially eliminate itself, would secrete something that would take away all of its components, leaving a void where it had been. And depending on where the cell found itself, only part of the cell's description is accessible. So I had the example where one set of these formed the bone-like structures, so they excreted the materials that form bone, and others excreted the material that form bellows. Similarly, cells, based on where they find themselves, will do things like assume an identity where they create a very stiff matrix, hydroxyapatite matrix, and form bone, or they will make actin and myosin and form muscle, or they will elaborate long cylindrical structures and find tree-like excrescences from the cell body that allow them to take in lots of information and become a nerve cell. All of these are things that cells are capable of doing during the developmental process. And so what you have is exponentially increasing cells are positioned in polynomial time. And I want to emphasize that if we think about this, this is a remarkable thing. You have a fertilized egg in humans. Over the course of 9 months, you end up from a single fertilized egg to something that has literally trillions of cells in it and is capable of organized behavior. And that's seen in many other organisms. If you were trying to position each of those trillion cells, even if you had only a fraction of a second to do that positioning, that would take far, far longer to do. So this is a way of manufacturing that is very different than the way we currently manufacture devices. And one of the critical things is also that the process varies so that even identical twins differ at the cellular level. And of course, if you have non-identical twins or who have just different siblings, they can differ quite significantly. Okay, and then when I spoke about evolutionary algorithms, I did so because I wanted to contrast the standard design approaches versus evolution. Here's a picture of the finches that Darwin described that he found on the Galapagos Island. He intuited, he didn't know this. Subsequently, Peter and Rosemary Grant did the studies to show that genetically this was actually correct, that these were originally colonized by one or just a few finches, maybe just a one mating pair that had come to the island. They were not originally native there, but they came over. There are various kinds of flotation rafts that animals can use to get to islands, or since they have wings, they can fly there. And what happened was in different regions of the islands, in some places, for example, where you had nuts that needed to be cracked, if you look at the top left, you see a very short, stiff beak. And in other places where there were, for example, cactuses, and you had to insert the beak into a very narrow area to get insects from the cactus, you ended up with a very thin, delicate beak. And all of these were initially from the original parents, but he saw that they were so different that they were classified as different species. This is one of the critical things that he recognized could be the basis for the origin of species. So what are the principles? Well, organisms vary. The variations allow an organism to survive more effectively in environment, and that surviving organisms leave more offspring. And variation is fundamental for evolution. It's actually the signal. It is not noise. So just as those strings of parameters I described have more, so to speak, descendants in the next generation if they have higher fitness, so organisms that are more fit in the environment leave more offspring and therefore come to dominate the population. And it's important to point out that during an animal's lifetime, process of selection acting on connections with the nervous system due to learning and plasticity allow for flexible changes in behavior. So there's a process of reprogramming that occurs in biological organisms during their lifetime because of the flexibility of the nervous system that they possess. Okay, now let's go back to our thought experiment and critique it just a little bit. So if we have devices that are subject to development and evolution, and I've shown you how the parallels, are they alive? And if not, what's missing? So I would not at this point claim that they're alive because, first of all, can the device harness the supply of energy? If you go back to the criteria that I presented earlier that describe some of the key things, can they harness the supply of energy? I suppose they could print photovoltaic cells, at least some of them that were going to be on the surface, and others could have motility that moved you towards a light source. Certainly there are currently robots that can find, if they have an infrared beacon, they can find where the plug is and plug themselves in. So there might be ways of setting it up so that it could find energy, but that would need to be carefully thought through. And how does the device obtain the materials needed to create its components and distribute them to its constituent elements? Now, in biological systems, that's why you have, for example, a digestive system, which after you've ingested material, which is not in its original form, again, if you photosynthesize, then you directly turn photons into energy. So that's why I mentioned the idea of photovoltaic cells. But if you don't photosynthesize and you're dependent on eating plants or other animals, then what you need to do is you have to find them, you have to ingest them, and then you have to transform them. And that's what digestive systems are for. They're breaking down all of the components of what they've ingested into molecular components that can be absorbed and then can go to the appropriate cells in order to be further used for energy and for raw materials. And you have a circulatory system that allows you to ensure that cells deep within the body are never too far from sources of oxygen. That's what your capillary system is for. So that allows you to deliver the key gases that you need, oxygen, to make the ATP and to excrete the carbon dioxide that you no longer need because you've used it up in metabolism, and continue to persist. And your lungs and your heart are all critical for pumping, for getting that air in, exchanging it appropriately with that circulatory system, the elements, the red blood cells I mentioned, and then distributing that through the body. So there are ideas from biology one could think about, and one would have to do some hard work to try to design things that could make those kinds of elements internal to a three-dimensional device like this. And that might make it closer to something that we would call living, because it would be more autonomous. Once we made one of these, they might be able to manage on their own. But again, this would take some additional work and thought. And finally, how could you effectively control all of the elements to generate adaptive behavior? So I suggested one way using genetic algorithms, but what might make more sense, and is what biological systems do for very complex behaviors, they have specialized cells, nerve cells, that are organized in particular ways and are capable of complex and flexible control. So let's talk a little bit about that and see again what we can learn from biology about that. So what I want to talk a little bit more is about biologically inspired neural networks and say something about control versus plasticity. So classical control techniques, as everyone in this audience probably knows, can be used very effectively for linear systems. You characterize the input-output, the transfer function of the peripheral plant that you're trying to control. You determine what are the control variables that you have access to. You look at the signals that you are getting back from the plant. You compare them to a particular error, to a particular goal that you might have, whether that's a fixed location or a trajectory. You take the difference, that error can be fed into the system and be used for corrective action using the control variables that can control the system. And especially if you have a linear system, this works very effectively. And again, there's some issues in terms of dealing with feedback and delays and so on. It's not trivial, but it can. This is a well-established field, and one can do remarkable things with control theory. Now what's happening to deal with nonlinear systems is people are starting to use artificial neural networks, and they're starting to show significant efficacy. So that's really very exciting. And it's of course worth pointing out that these are inspired by biological neural networks. However, they have some significant drawbacks. They do require very large training sets. Once you get one that works, you just have a set of connections, and it's really hard to understand what those connections mean or imply or how they work. And these can be, in some ways, quite brittle. People have shown that you can, for example, on the sensory end, you can get networks that are well tuned to particular sorts of things. You make a small change, they no longer recognize those objects. And that small change is something that would not bother a biological neural network or visual cortex. So something is missing, and again, it's very hard to figure out what the differences are and why they are that way. In contrast, it's important to recognize that even animals that have relatively small numbers of neurons are capable of rapidly learning from experience and can flexibly alter their behavior. So we have 10 billion neurons, that's a lot, and trillions of connections, hundreds of trillions of connections between them. But even an insect, which has about 1 million cells, can do remarkable feats and can figure things out. And the sea slug that I love and study has about 20,000 neurons, and it's capable of feeding and locomotion and mating, and it can learn, for example, that food is inedible. And animals, from the time that they come out of whatever enclosure they're in during the after development, whether it's an egg or they're born from a mother, are usually able to do significant numbers of things on their own. Once Aplysia hatches, it's a free-swimming veliger. It has these velar lobes, and it can be a filter feeder. And then once it transforms on Laurencia, the seaweed it loves, it becomes a little slug, and it immediately knows how to feed on seaweed. Doesn't have to have any training. It can learn, however, which parts of the seaweed are inedible and which are edible, which are very important for its survival. So the question this raises are, what are the features of biological nervous systems that allow them to be so effective? And that's what I'd like to spend a little time talking about now. Okay, so these features are well known to neurobiologists. Many people who have an engineering background, and the last time they confronted biology was perhaps dissecting a frog in high school. And after looking at a textbook filled with lots of weird names and complicated structures, all of which we're supposed to memorize, they closed the textbook and said, "I'll stick with math, thank you." And I can actually appreciate that viewpoint. But I'm going to just give you a high-level description of what it is that makes real neurons so effective, and perhaps reinterest you in learning more about the biology. Starting with Pitts and McCulloch, the idea was that neurons were just on-off devices, and they basically could summate the inputs, and they could more or less then make a decision whether to be active or not active. And in fact, in artificial neural networks, you've got the various weights coming into a neuron, and a neuron is represented. It's a little bit more sophisticated than just 0 or 1. It's a sigmoid function, which would represent a continuous range from being off to being on. But that's actually a very, very simplified caricature of what neurons actually are. Neurons are actually each complex dynamical systems. What do I mean by that? An individual neuron has in its membrane a series of channels that allow certain ions to come through, and these channels can be gated. They can be opened or closed by either the voltage across the membrane or chemicals that bind to the membrane. The consequence of this is that neurons can actually generate activity in the absence of any other inputs. They can be spontaneously active. They can generate rhythmic bursts, and those rhythmic bursts can be very complex. They can fire fast initially and slow down, or they can fire slowly initially and speed up. And they can fire irregularly, and they can be silent. And even more importantly, they can have multi-stability. That is, as a function of the inputs they're getting, other chemicals around them, they can shift from being completely silent, for example, to rhythmically bursting and back. So this is the characteristic of what we call a dynamical system. It's something that shows complex processes over time. And it's certainly not just an on-off device. Now, that complexity is further enhanced by the shapes of neurons. Neurons have a wide variety of incredibly remarkable shapes, very complex shapes. Ramon E. Cajal, working in his kitchen, this is before the era of NIH grants, using the Golgi stain, would impregnate these blocks of tissue. And then he was a very good artist, and he would draw these phenomenal pictures, which, you know, if you hadn't subsequently seen this, you would think he was just hallucinating, because they have all these elaborate structures. Neurons have very different shapes. Now, the reason that's important is this: where an input comes in. So let's say this is the central part, the cell body of a neuron, and the decision point as to whether it's going to start to fire. Let's say it's down here. So if an input comes into one of these little fine branches called dendrites, if it's out here, it has to travel electrically down to here to activate. But if it comes in much closer, like here, a much weaker input can generate the inputs. So a whole series of very strong inputs can come at the edges at fast frequency and turn it on, or a few well-placed inputs can turn it on. It's the difference between, for example, voters in a democracy trying to set the direction of their country versus someone who has the ear of the executive and can immediately get something done because they can just talk to them and get them to approve something. So a more general way of saying it is that neurons respond to complex spatio-temporal patterns of input. So that's rather different than just summing up weights. Okay, and the neural connections themselves, the connections between neurons, have complex dynamics. And depending on how they work, they can be private. The chemical that's released can just talk to one other neuron in the synapse within the synaptic cleft, and that's like a private telephone conversation. Or like a peptide can be released and can affect not just the adjacent neurons, but some neurons in the vicinity over a longer time scale and a longer spatial scale. That's like somebody having an active conversation on the bus, something I always try to ignore, but I can't so easily do because they want me to know about what they're doing for dinner and what so-and-so said to them yesterday and so on. They feel the need to share. In the nervous system, this is actually rather useful, whereas on the bus, if you're trying to read and think, it's somewhat annoying. But unless you're really interested in overhearing other people's conversations. And then finally, they can act at very long groups, and large groups, and very long distances. They are almost like a radio transmitter, where if you're tuned to the right channel, you can get the message, you can get the signal, you can hear the music you're interested in or the news from great distances. You just have to have the receiver tuned properly. And that's like hormones, which as long as there's a receptor on a cell that can bind it, the cell can respond to that message and act on it. That's very different than the kind of uniform connectivity pattern that you see in artificial neural networks. Another key thing is that what the artificial neural networks are doing is they're trying to adjust the specific connections as a function of experience. But one of the issues in nervous systems is there are ways of tagging which of the connections are important as a function of the recent experience, and those are the ones that get changed. Not all the connections, but the relevant connections. In some studies I've done with my colleagues in the slug, when we look at the connections that are changed after you teach an animal that food is inedible, those connections that are important for rejection behavior are actually strengthened between the sensory and motor neurons, and the ones that aren't are not. So you can have selective changes in plasticity. Another important issue is, again, the architecture now for artificial neural networks is you have at least 3 layers and maybe more, but they're just layers. And then from each layer, there's a huge number of connections to the next layer. That's not how neurons actually are set up. There's a complex architecture, and that architecture gives rise to things like cortical columns or the cerebellar architecture that's able to transduce information in very complex ways. And so architecture matters. It's very important. And another important issue is that the control can be shared with the periphery and the environment. So for example, if you watch an eagle, at some point as it's looking for prey, it may be riding a thermal. It's not flapping its wings. It's not doing anything with its body at all except riding that thermal. So its nervous system is being used to look for prey, but it's actually not flapping its wings at all. It's using the environment for flying. And then of course, if it sees something interesting, it starts to flap, it drops rapidly, and goodbye to whatever it is that it's cited. Similarly, if you have a heavy leg, this is one of the points that Greg made in his excellent presentation yesterday, then at the onset of swing, if you have a heavy leg, the impulse to the leg is sufficient through the inertia of the leg to keep that swing happening. And in fact, you need another impulse on the opposite set of muscles to brake and slow that, because inertia otherwise would keep you going too far. So you're sharing the control with the periphery and with the environment. And control emerges, and this is the key point I want to make and leave you with, that control in the nervous system emerges by shaping neural dynamics, peripheral environmental dynamics, whose initial properties are set by evolution and development. And one of the ways that we are trying to take advantage of this, you'll see presentations about this, is by creating what we call synthetic nervous systems. This is work that Nick Shazinski and Roger Quinn have pioneered, and there's several presentations in this meeting, which I encourage you to listen to, where the neurons have some internal dynamics, they have interesting architectures. They're still simpler than real biological neurons, but they're a step closer to the way real neurons work. And therefore, I think that's one of the reasons why one can do some very interesting control with them that one can't do otherwise. Let me briefly talk about directions, the other direction. This is all talking about how one can try to create artificial devices or simulated devices that are biological alike. But what's going on right now is in the biology side is absolutely astonishing. Synthetic biology now can create designer genes cells, organs, and even organisms. That's a realistic possibility. There was a really nice article in the New York Times Science section a few weeks ago about the new uses of CRISPR-Cas. CRISPR-Cas allows you to snip out pieces of DNA and insert other pieces of DNA. So if you want tomatoes to make a particular kind of compound they never make naturally, you can get them to do that for you. This very controversially, someone actually tried to do this in human embryos, a Chinese investigator who now I think is in prison, to try to create HIV resistance. But the possibility of changing these things and engineering them is now becoming possible. And it's very interesting because if you work on the germline, things that affect the manufacture of sperm or eggs, then it's a hereditable change. It's not just a change that will be in the lifetime of that organism and then die out with that organism. All of its descendants will have that change. And another direction that people are going is to try to simplify down things like mycoplasmas that are very, some of the simplest organisms, and see, can I eliminate many, many different elements and create a minimal cell that could then serve as the basis for creating novel artificial cells. And people are also working very hard at making excellent progress on developing simulations of these minimal cells, which allows you to begin to explore how they'll behave in response to both internal and external changes. Now, before I end, I just want to point out that all of these things I've been talking about have very important ramifications that go beyond the excitement of just let's make something cool and new. As we know, for example, the Great Lakes were recently, some years ago, the quagga mussel was introduced probably from the ballast of ships that use the Great Lakes. And these quagga mussels secrete this gooey stuff and they outcompete all the natural mussels and they clog the waterways. And they're an invasive species. They weren't there originally. Now, as things are warming up, species that would tend to die out through the winter and then couldn't go beyond certain regions are now beginning to evade new regions. And this is all happening naturally. But as we see, when that happens, that can have massive disruptive effects. We've just been experiencing and are still experiencing how a pandemic, a virus, which was not, I believe, released by humans, it was probably jumped from an animal to a human is the most likely explanation based on the evidence. But they show how new variants of bacteria can have major impacts on human populations. So the problem here is that if we succeed in creating artificial self-replicating devices that can modify themselves in response to environmental conditions, they could have profound social, economic, and political impacts. At the same time, therefore, that we work to develop these devices, I think it's very important to make sure that part of our teams involve ethicists, and we must encourage regulation to ensure that these devices are properly controlled. It's a little bit too late when the thing has escaped from the laboratory and is now devastating the population. You go, oops, I didn't mean that to happen. We need to do some work in advance of that and get ahead of it. And then if we make this part of our concerns in the years to come, then I do want to end on a very positive note, because some of the things I'm discussing, and I hope some of you actually got excited by this cell machine that I described and its potentialities, we have the possibility of reaping remarkable benefits. And we could avoid the considerable harms they might otherwise cause. So I'd now like to wrap up with a summary and conclusion. The first point is that cells and organisms can be thought as complex molecular machines. That's a term that you'll find people using. So that answers one of the questions that I raised. Are living things machines? People call them molecular machines, but they're a very special type. They constitute a network of processes that produce the key components necessary to maintain the operation of the network as a unit. They're able to assimilate energy, all the other things we talked about. But the key thing is that once you set them in motion, they can, so to speak, stay alive. They can take care of themselves. You don't have to plug them in. You don't have to change their batteries. You don't have to make sure that they're positioned in such a way that they're receiving the appropriate signals. They just do all that stuff on their own. There's a fundamental autonomy and self-containedness that seems to be critical for these kinds of molecular machines. I pointed out, I hope this, by making it very concrete and painting these pictures for you verbally, that concepts from evolution and development could be used to create artificial devices that could very rapidly self-assemble and could be updated through creating and evaluating many variants, either of the actual devices or through simulation. And I talked about how synthetic nervous systems, by incorporating many more features of biological nervous systems, may make it possible to control complex behaviors and adjust them effectively over time. I also pointed out that new advances in synthetic biology and what's called artificial life may soon create devices that really almost completely blur the border between the living and the non-living. And I concluded by emphasizing that the major impacts such devices can have socially and politically require thoughtful consideration of ethical concerns and careful regulation. Now I'd just like to thank several people. Many of the ideas that I'm talking about, especially focus on autopoiesis and genetic algorithms, were done with my colleague and friend, Randy Beer. Roger Quinn and I have been working for many years to create biologically inspired robots. And I don't know whether he's now going to create a new branch of his lab to create cell machines. I wouldn't put it past him. I've also had the privilege of working with Victoria Webster Wood, who you'll be hearing from on Friday, on developing biohybrid robots, and with Greg Sutton developing concepts of understanding how biomechanics affects energy distribution and also neural control. I've had a long-standing collaboration with Peter Thomas, who is an applied mathematician, and we have used some of the things I talked about, some of the simpler models of these neuromechanical systems to understand more about robustness, flexibility, and how to deal with real-world conditions like changing loads. My colleague Abraham Sussman I mentioned briefly because I talked about the learning work that I've done with him. And the original model of the neuromechanics was done with Kendrick Shaw and was developed further by a postdoc that I shared with Peter Thomas, David Lidle. A vital member of my laboratory for many years has been Jeff Gill. Much of the data we've gotten is from him, but Jeff, in fact, started with an interest in autopoiesis, and I hope that he continues that interest. Some of the students who got the data that I mentioned for the learning project, which Shlomi Tom and Itai Horowitz. Another critical person in my lab is Jade Yuan, a graduate student who's been getting very important data for our understanding of the dynamics of the neural network we study. Yanjun Lee will be giving a talk. He is co-advised by Vicki Webster Wood, Greg Sutton, Jeff, Roger, and myself on a artificial neural network, a synthetic nervous system for controlling feeding in Aplysia and showing that it can be multifunctional. I'm very grateful both to NSF and NIH for their funding of my work. And with that, I'm happy to take any questions that you may have. Thank you for your attention.
Thank you very much, Chiel. That was very, thought-inspiring. It's I always have questions, but we'll leave it up to the audience.
Right. Well, I think Jeff has summarized it by saying, "I, for one, welcome our new living 3D printer cell machine overlords." Thank you.
Questions? Paul.
Yes, Paul.
I knew Paul would have questions.
Yeah, look, okay, this is really great thought experiment, right? And great analysis. But the theory of evolution is also complexifying with the day, right? So now we are in sort of the extended modern synthesis, where also new layers of control loops are brought in, right? You have evo-devo, right, where you also start to have now environmental impact on transcription, but also beyond that, niche construction, parent selection, cultural evolution, which are now all brought in as additional levels of organization, feeding back in the end into this whole notion of the change that might be due to heredity. So in the proposal you're making, Hillel, how would you then include these additional levels of control loops? Of course, we can say, oh, it's accounted for, we can do it, right? But that also would require another level of engineering that would go beyond 3D printing the physical instantiation of the exemplar, right? Of the phenotype. So how are you going to solve these additional layers? Because otherwise I cannot get started.
Well, first of all, I think it's important to recognize that even though these various aspects are important, there are simple organisms that are capable of perfectly well replicating themselves, like bacteria, that don't seem to use the higher levels at all. And they're nevertheless capable of doing some very useful things. So you don't have to have these things necessarily to get started. That's what evolution shows us. The second point, though, I take very much what you're saying. The issue is that if you talk about these self-organizing elements, like these cell machines, I commented very briefly that the lineage and the position determines what you do next, what part of your self-description is activated. That's what happens in development. So if you wanted to incorporate into that certain epigenetic factors, such that certain parts of the self-description are unreachable as a consequence of something that has happened in the environment, or they are activated because of something that's happened in the environment, I don't think that's beyond the reach of the idea that I suggest. Now, if you have these we'll call them artificial multicellular devices interacting, and they actually have synthetic nervous systems, the possibility of fairly complex behavior emerging from within those groups is certainly possible. Beyond that, I don't really want to speculate, but I just say that the history of evolution is such that you can get increasing layers of complexity starting from great simplicity. That's what the fossil record seems to suggest. And therefore, I don't think that it's beyond the reach. But I agree with you that there are many steps that need to be made, even to implement what, and this is why I was self-critical, even to implement what I suggested as a thought experiment, dealing with the question of getting the energy, of distributing the energy and distributing the raw materials is non-trivial, would take a great deal of work. But if we start with something simple, the beauty of both evolutionary and developmental processes, as I tried to point out, is that you are not starting from scratch. You can build from good initial conditions and make things even better. And that, I think, is really the profound message.
But now in this Cornish-Bowden review that you started out with, which is actually a very useful paper.
Yes.
You come out with a checklist, right? But also actually they do emphasize this core concept of autopoiesis, of sort of self-maintenance, quite a bit in that article. So I also could imagine you would have taken that as your core feature, because that sets another set of criteria. So why did you move away from that and went for a fuller list?
The reason I did that was because one of the things that was so nice about that particular article is they talked in detail about the hypercycle approach, the MR systems, the autocatalytic systems, and the chematon. And they pointed out some of the defects that are found in each of the different models. And what I was pointing out was not so much a list as a set of desiderata for a theory, whatever that theory might end up being, that one would want to ultimately develop. So I actually have been very influenced by the concepts of autopoiesis. My hesitation with it has been, and this is why I found that article so very helpful, is that in terms of actually creating simulations, I mean, Varela tried to do that in the 70s and was able to actually get something to work again in the 90s, but it was fairly primitive. And one of the things that those reviewers actually have done, they have created very detailed and much more realistic simulations that have some of those properties. And they've been inspired not just by autopoiesis, but by the other ideas. So I wanted to give credence to that larger group of ideas rather than just focusing on one.
Okay. Great. No, thank you very much. This was a great way to get started. So thank you very much.
Thank you for your kind words. I appreciate that.
And Nick, you had a question. You seem to have dropped your hand though.
Yeah, in the interest of keeping everything on time, I'll catch up with Hillel later. But it was a great talk, Hillel. Great scope, great way to get us started, like Paul said. So thank you.
I appreciate that. And again, if someone is inspired by what I've said to actually start trying to do this, I'm sharing these ideas. And I freely emphasize that they were inspired by the biological system, which is why I'm giving them away. But if you found it useful to talk to me about them, if you tried to do this, people have worked with me in the past on biologically inspired robotics and other things like that have found it useful to get some of my insights, and I would be happy to be involved in those discussions. So I guess I could leave it at that.
Okay, but let's thank Hillel again for a beautiful presentation. It was great. So we have a break until 20 minutes after the hour, and we'll see you again in about 17 minutes.
See you all.